is on your protein shake. That is a true statement. So one person was like, no, shape doesn't matter. Does shape matter?
Yes.
Because form is always related to function. So if we change the shape, then our protein can't work anymore. Okay, some globular proteins act as molecular chaperones. They help other proteins achieve their shape so they can be functional, so it puts them in the right three-dimensional shape. We're not going to do a ton with molecular chaperones this semester. In the chapter on, I believe it's chapter 16, on the endocrine system, you'll have to learn a lot more about molecular chaperones because some of your hormones require chaperones to get to their destination. Otherwise, they're like, yeah, I'm not going in water. I'm staying out here because they're hydro-wet. Phobic, they're non-wet. Polar, they're non-polar, they're hydrophobic, and they're not going in the water. So molecular chaperones do play a very important role in the metabolic processes and in the metabolism that happens in your body. Okay, now let's talk about enzymes. We're going to take a minute and just really focus on enzymes. We'll talk more about enzymes as we go through some of the chapters this semester, and you're going to hit them really hard next semester, especially when you start looking at digestion. You'll look at lots and lots and lots of enzymes. We'll do a little bit like ATP synthase this semester, and some of the more prevalent cool ones, and then you'll get to learn all about their cousins next semester. Enzymes are always going to be globular proteins, and they catalyze reactions. A catalyst is something that makes something else happen faster. So it is, in our case, an organic molecule of protein that makes a reaction in your body happen faster. And they can make reactions happen up to 1,000,000 times faster than they would otherwise happen. In other words, they can take it from this isn't going to happen to, oh yeah, it's like that. And that's important because I can't teach you, right? We have to have some way to increase the speed of reactions because we can't keep up living things because it kills them. Enzymes tend to be very, very specific. Most of the time an enzyme does one thing. It does only that thing, and it does it very, very well. Now, there are some times where an enzyme will make a reaction go one way and also be able to make the reaction go the other way. But most of the time, it just makes the reaction proceed in one direction. And my favorite example, of course, when I first start talking about enzymes, is the first enzyme that is going to be used inside of your body. And that is amylase.
Oh, yeah.
Amylase is an enzyme found in your saliva that breaks down a polysaccharide. That's a big sugar called amylose. O tells us it's a sugar. A tells us it's an enzyme. All of the sugar breaking down, so anything that breaks down polysaccharides or disaccharides is going to end in ACE. Now you're going to have a whole bunch of enzymes that don't follow the ACE rule, like pepsin, but we're not going to worry about that in a minute. Amylose, by the way, is the name of the polysaccharide that you probably call starch. I know. Why are you giving it such a big name? Because amylose is the scientific name. It is the sugar name of this compound. So far, so good? Yes? So amylase only breaks down amylose. It doesn't break down any proteins. It doesn't break down lactose. It doesn't break down anything else, just amylose. It is specific. They act on substrate. Again, the substrate is the reactant in an enzymatic equation. And enzymatic reaction. That's the left side of our equation. Most enzymes are holoenzymes, which means they are going to have two parts. They have an enzyme, which is the protein portion, and then they have a cofactor. That's something that has to bond to the enzyme to make it the right shape to do its reaction. And we use cofactors in our bodies to activate enzymes, to make them so they can do their job. Because we want to be able to turn those guys off and on. We don't want them always running around willy-nilly doing their thing. We want to have some control, and the cofactor allows us to have that control. If the cofactor is a vitamin, then we would call that cofactor the coenzyme. Are vitamins important in your diet?
Yeah.
Yes, because they act as coenzymes that allow your enzymes to function properly. If you don't have enough of your vitamins in your diet, no matter how many calories you have, you will still be malnourished. So you can literally be morbidly obese and be malnourished. Because malnourished means you don't have the right foods. It means you're not getting the proper nutrition. So we want to be careful that we're eating the right kinds of foods in the right amount so that we can stay healthy. I eat all the right kinds of foods, and then I eat all the wrong kinds of foods. That's my issue. Okay. Let's look at activating my enzyme. The energy of activation, and this is a physics and chemistry thing, but we need to know it for biology too. The energy of activation is the amount of energy required to start the reaction. All reactions have energy of activation. You've got to get them going. Normally, if you're looking at a reaction happening in a test tube, you can add enough energy to start a reaction or prime it in a couple of different ways. One, you can heat it up. How many of you have had chemistry? Have you ever, gone to chemistry class and you boil something to make the reaction happen? Yeah, you put a little water bath on a hot plate, make it happen, or you hold it over a little flame and make things happen. We can't do that in our body. The other way you can make a reaction happen faster, and we do this in chemistry too, is you can stir it up or shake it. Have you ever had a chemistry lab where you had to swirl something? Or you use a little vortex and you put it on there to go like that?
Yeah.
We can't do that with you. What will happen to you if I just shake you like this? Like your brain will be all knocked around in your head. That's not a good thing. So we have to have other ways of activating our chemical reactions, and we use enzymes to do that. So we're going to look at the effects enzymes have on energy of activation, the amount of energy required to get a reaction to go. Starting with this is an example. This isn't an actual reaction. It's just sort of a what is kind of thing. Let's look at our first reaction. My reaction on the left side of the equation, they're over here, takes this much energy to make them react, and then my reaction will proceed and I will make my product. We're looking at the rise and fall of the energy. I had to put in quite a bit of energy. On the right side, I'm looking at what happens if I have an enzyme. I still have to put in energy, yes? But I get to use way less energy to get to my product. Are you guys familiar? There's this game that people play called baseball. Have you heard of it before? You thought it was me? Okay, in this game called baseball, you have a guy called the pitcher, and he throws a ball at a guy called the batter. I don't know why the batter just stands there, but he always does. The pitcher throws the ball at him. The only defense the batter has is this thing called a bat. Get it? Batter. The batter has the bat. Okay, so the batter's like this, and he is going to hit the ball. The amount of energy he puts into hitting the ball is the energy of activation. And then the ball is going to go and go and go and go and go, and sometimes it goes over the fence. That's called a what? Home run. Woo-hoo! So, not a touchdown. No. That's a home run. You got the wrong game and you said touchdown. That's a different game. So the ball goes over the fence. He has a home run. He has to put in a certain amount of energy, right, to get that ball to go over the fence. So far, so good? Yeah. Okay. What if I take away the fence? What if I knock down the fence? Will he have to hit the ball as hard? No. Taking away the fence is what the enzyme does. It takes away the fence. So he doesn't have to hit the ball as hard, and he still gets a home run. That's my example for my kinese guys. Kinese guys in the back, do you understand it? Energy of activation, got it? Okay. If you don't understand baseball, and I know you're kinese too, if you guys don't know baseball, you are in the wrong major. I'm just saying. Do I have any baseball players this semester? Okay, let's talk about the steps of enzyme activity. Starting with, you have your enzyme, the substrate bonds to the active site. Remember, the substrate is the reactant in an enzymatic reaction. Bonds to the active site. In this case, when we get to the end, I'm going to make you tell me what my substrate was based on bonds and stuff, so be ready for that. When the enzyme is bound to the substrate, we call that, wait for it, the enzyme substrate complex. Okay. So on your test, when I say, what is the combination, or what do we call it when the enzyme is bound to the substrate? What is the resultant molecule, or something like that? And one of the choices is enzyme substrate complex. Some of you will be like, oh, that has to be a trick. It's not a trick. That's just how much we are not creative when we name things in biology. We are really direct about it. It's either what it is, what it does, or who figured it out. Those are the ways we name everything. Okay. Next, the arrangement of my substrate is changed so that a reaction will occur. In this case, we're forming a bond and removing water when you test. You guys, we know this one, right? What do you call it when you form a bond and remove water? Dehydration or condensation synthesis. If you didn't know you needed to know that, you might want to write that down real quick, because I promise you got that on your test. After that, the enzyme is released too. Check it out. It looks exactly like it did to start with, right? So we can start over. And the product is released. Oh, my substrates are now held together by a peptide bond in my product. What were my substrates? Amino acids. How do we know that? Other than it says. How do we know by looking at this picture? Amino acids are the only thing held together by peptide bonds. Remember, that's the nitrogen to carbon thing? So anytime we join amino acids, we make a peptide bond. Yay. That wasn't subtle, right? You know that for your test? Oh. Guys, guys in the back, do you know that for your test? Okay, just making sure, because I know some of you got off on baseball, and you might be thinking statistics now, so I'm making sure you're still on the page with us. Okay, let's talk now about nucleic acids. Nucleic acids include DNA and RNA. DNA stands for deoxyribonucleic acid. I'm going to give you a hint about DNA. The sugar in DNA is called deoxyribose. And it's a nucleic acid. Got it? So it's deoxyribonucleic acid. It's named after its sugar. RNA is ribonucleic acid. Wait for it. Its sugar is called ribose. Get it?
Yeah.
So deoxyribose, we just took off the left foot of the oath, and ribose, we just took off the left foot of the oath, and there's the name of our nucleic acid. I think that's pretty cool. That's one of the fun naming things. It means you don't really have to know very much to figure out what something is if you know the name of it in biology. Okay, DNA is a double-stranded molecule. It's found in the nucleus of the cell. It is a double helix. That means there are two molecules twisted together in it. It encodes all of the information necessary for life. You probably already knew that DNA genetic material, right? We've known this for quite a while. The whole Chargaff law and all of that plays into it. We're not going to cover that in anatomy, but in Bio 1 we do it, so probably you have already done it. It is important. Just honor DNA for a moment. Okay, RNA is also important, and RNA was probably evolutionarily the first genetic material. It is single-stranded, and you make it inside of the nucleus and then transport it out of the nucleus and use it in the cytoplasm of the cell. The RNA is where you directly are coding for your proteins. And proteins are the structural and functional molecules of your cells. So the RNA takes the message out so that the cell can do the work that the DNA is coding for. There are 5 nucleotides that make up nucleic acid. The nucleic acid is the polymer. That's the big, giant, enormous, like 6 feet long molecule that DNA can be. The nucleotide is the monomer. So you have whole nucleotides floating around inside of the cell. All nucleotides have 3 parts. They have a phosphate, a sugar, and a nitrogen-containing base. You put together your nucleotides way before you're going to make your DNA. They're kind of just floating around and they're waiting to be used. So you don't just add a phosphate to DNA, you add a whole nucleotide. So someday when you take genetics, you have to know that. And he'll make you be able to draw this. I won't make you be able to draw this. We're just going to kind of look at this, but we're not going to draw this. If you want to learn to draw, then you may. I know, kids, you guys love to draw stuff. So if you want to learn to draw, then go right ahead. Okay, so we have 5 nucleotides. We have our adenine. Adenine is a purine. Purines have 2 rings. We're going to look at some pictures of those in a moment. Purines are double rings. Then you have thymine. Thymine is a pyrimidine. Pyrimidines have 1 ring. The thymine is only found in DNA, not in RNA. Then you have guanine, which is a purine. How many rings does guanine have? 2. And you have cytosine, which is a pyrimidine. How many rings does cytosine have? 1. Oh, I always remembered that. Well, actually, I just remembered it. But the way that I usually tell people to remember it is the bigger word has the fewer rings. So kind of, I guess it's like a chihuahua. It thinks it's tough. The purine gets 2 rings because it's got the little rings. Anyway, uracil is a pyrimidine found only in RNA. The adenine always pairs with the thymine. And the guanine always pairs with the cytosine in DNA structure. I just always remembered that the round letters go together. There are some other ways I have written to remember, but they're dirty, so I'm not going to tell you. You want to know? Okay. You want to know. Adenine pairs with thymine. T pairs with A. And we used to call movies that have nudity for no reason T and A movies. If you don't get it, you can ask someone to explain it to you later. Anyone my generation will be offended when you ask the question. So they'll be like, what is teenage? Oh, my God. Oh, okay. And you'll embarrass your mom if you ask her. Okay. Let's take a look. Adenine here with its 2 rings is bound to thymine with its 1. There's 2 hydrogen bonds holding those. Oh, I can put it up here. There's 2 hydrogen bonds holding those together. The cytosine and guanine paired together have 3. Now, what's going to happen? Oh, okay, let's go ahead and do the question first. I forgot the question was next. What are the monomers of DNA? Are they called nucleic acids? you're not. We gotta go. Right? Okay, 3, 2, 1. Everybody should be in. Let's take a look and see what you came up with. Yay! The nucleotides are the monomers. DNA is nucleic acid, which is a polymer. Amino acids are found in what? Proteins, not in nucleic acids. DNA is not made of amino acids. And DNA, of course, is a ginormous, I keep telling you, like 50 long. It's a huge, huge molecule. It's not a monomer. It's a polymer. Okay. So let's look at the big picture structure of our DNA. Adenine, as I said, pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. And that's what I'm showing you here in my double helix diagram. You can see that they kind of snake, the 2 strands kind of snake around each other. They have a phosphate sugar backbone, which means that this part goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. And the bases stick out and make the rungs of the ladder. It's kind of cool, right? Let me tell you what's so cool about this, because you may not have recognized it yet. You'll figure it out later, but I don't want you to have to sit around thinking about it all day, because you have other stuff to study. If you look at the adenine with how many rings?
2.
2. And the thymine with how many rings? 1. So you have 2 plus 1 is 3. If you look at the guanine with the cytosine, the guanine has how many? It's a purine that has how many? 2. And the cytosine is a pyrimidine. It has 1. So 2 plus 1 equals 3. So as long as a purine goes in the pyrimidine, we'll have 3 all the way down, and the diameter of our ladder will stay even. If I had a pyrimidine with a pyrimidine, I'd only have 2, and if I had a purine with a purine, I would have 4, and my DNA would go like this, right? Now let me ask you something. Would you get onto a ladder that goes like this? Why not? It's unstable. So the constant width of the strand of DNA makes it more stable. Isn't that cool? Now you know something you can talk to your friends about at lunch. It's true. Guys in the back, you can talk about climbing the stable DNA ladder. Okay? Okay. Let's talk now about ATP, or adenosine triphosphate. And why it's called that? It's called that because it has adenosine, which is the ribose sugar plus the adenine, adenosine, plus 1, 2, 3 phosphate. Yeah. When adenosine is found to only 1 phosphate, we call it adenosine monophosphate, or AMP, and we'll use that molecule in a few things later in the semester. When we're looking at 2 phosphate, it's adenosine diphosphate, and when we're looking at 3 phosphate, it's adenosine triphosphate. The 3rd phosphate bond, this guy out here, is a very, very high energy bond. And your cells use the energy from that bond to start other reactions. They use that as their energy activation. What we call that then is the energy currency. So ATP is the energy currency of the cell. It's the kind of energy your cell can use. Your cell cannot directly use sugar energy. There is too much in there. If your cell were to try to use that directly, it would be like a little explosion going off in the cell. I know. A little tiny, tiny explosion in a little tiny, tiny cell. Take down the whole building. The whole cell would just right down here. Okay. You break the phosphate bond out here by hydrolysis, of course. That's how we break all of our bonds. Yes, it's hydrolysis. And that releases that energy. And then you can use it for other stuff. Let's look at what ATP energy can be used for. And I have a little issue with my arrows over here on the left, and we're going to talk about that in a second. So get ready to change the arrow. ATP energy can be used for transport work. That literally means moving molecules This little I means it's an inorganic phosphate group. It means that it doesn't have carbon in it. What's the deal with this arrow? Why is that wrong? What do I have to do to get it to go back to ATP? I have to take the water back out. But this picture was in your book, and it shows the water going in no matter which direction I go. This has to be flipped over. It has to show the water going where? Out. So make that change in your notes so you know we're not putting water in to make bonds. We're putting water in to break this. Right? Because that would be like that wouldn't work. Okay, what is ATP used for? Chemical work, transport work, mechanical work, or all of the above? What is ATP used for? Chemical work, transport work, mechanical work, all of the above.
Would you first press yours or when you press this, not on?
Oh, you just press the number. You don't have to press the
Okay,
turn it on.
3,
Press your answer. So that's
2, 1. Let's see what we came up with. Almost everyone got it. That is all of those. ATP is used to do all of the kinds of work that your cell does. So any work in your cell requires what? ATP. What was the name of the energy currency of the cell? ATP, adenosine triphosphate. How many phosphate groups does ATP have? 3. That is the end of the chapter on ATP. Okay, on chemistry. I am going to dismiss you because I do not want to start cells with only 6 minutes of class. We will run as fast as we can on Wednesday.